Welcome to CE, Civil Environmental Engineering Seminar. Today we have Professor Kathleen Treseder. She's here to present her work on fungi. She's a professor as well as chairs in Department of Ecology and Evolutionary Biology at the University of California, Irvine. She has a PhD at Stanford. Her work mostly on fungal biochemical processes. She uses molecular techniques, nanotechnology, as well as isotopes to understand how fungi behave different way in ecosystem or how they respond to different climate change. So we are really excited to have her here. And again, I have a lot of interest on that. So it's all you now, Professor Treseder. Go ahead and start presenting. And students, you can start, you can mute yourself if you, just in case.
All right, well, I'm so happy to be here. Thanks so much to Sanjay and Shaylee for inviting me and talking with me. Really exciting about the work that people are doing in your department, especially with bioremediation. So I'm hoping that what I talk about will be relevant. So today I'm going to talk about how fungi might kind of control how ecosystems respond to climate change. And, I did not start off studying fungi. I studied plants when I was a PhD student, but I switched over to fungi for my postdoc and for the rest of my career because I learned how important they are in ecosystems and also how understudied they are just in general. So fungi are really important because they perform a number of services in ecosystems. One is like bioremediation. So they can break down all sorts of compounds. I think just about any sort of compound out there that has carbon in it, there's likely a fungus somewhere that can break it down. There are fungi that can break down concrete even. It's amazing. So they can take up, kind of hazardous compounds in the environment, break them down, convert them to less hazardous forms. Along those same lines, they also break down dead material in the environment. So like dead wood and other plant parts, and they break down dead microbes and other things too. And as they do that, they release CO2 as a byproduct. And of course, CO2 is greenhouse gas. So that process they're conducting is part of warming. And so we need to keep track of that process in order to adjust our policies as we respond to climate change. Another reason that I like to study them is because they're so beautiful. So here's a picture of a fungus that actually glows in the dark. It's called foxfire. And so the people who are studying it, we don't know why it glows in the dark. It might be to attract insects, but it is beautiful for sure. Okay, so this is part of my lab, and in my lab we have a common mission. This is what gets us out of bed every morning, and it's to improve predictions of climate change over the next century or more. And the reason that we do that is because if we have these predictions and they're good predictions, we can start making public policy to either deal with this predicted climate change or try to prevent that climate change from happening. So everything that we do in our lab, one way or another, fits within this mission. Okay, and so one of the things that I want to point out is, we talk a lot about climate change and people are concerned about our future climate, but climate change actually is right now harming people, especially in tropical and arid regions. All right, and so one example here, this is a farmer from the Central Valley. I think a lot of you know, we just came out of a very severe drought in California. It lasted 4 years. It was the most severe drought of the past 500 years. So that's climate change. And by the end of this drought, quite a number of farmers in the Central Valley had lost their farms simply because they didn't have water to irrigate their crops. And the ones who lost their farms were the small farms, the family farmers. And so they've had to find whole other occupations to feed their families. So this is a threat in arid regions where we might be seeing more drought under climate. It's also a threat in warm regions like the tropics, where organisms might be already at their limit for their climate tolerances. And so this is something that's happening right now. There's really an urgency here. So we want to know like how to help people now and in the future that are experiencing these situations. We want really good predictions of how climate will look over the next few centuries and where, like what regions of the world will look like. Okay, so in order to do that, people use, it's called these Earth system models. These are big models. They're basically climate simulators. They run on supercomputers. We have some here at UCI. We also have some at UCLA that run. We include in these Earth system models everything we know about climate and how it works. And then we add CO2 from human activities. And then we track to see how temperature changes. Okay, so this right here is a graph showing a range of predictions from different Earth system models of future climate. And there are 12 Earth system models here in these predictions. And so I'm going to explain what's going on. So here along the x-axis, we have year, starting with the Industrial Revolution and going out a couple centuries in the future. Okay, so here on the y-axis, we have global surface temperature change. This is degree C compared to the global temperature that we're experiencing right now. Okay. Now here we have historic measurements, and then these right here in the yellow, these are the projections. Now you can see here that there's a big variation in these projections, and that is because the models do not agree very much at all on what future climate will look like. And this is important because the climate that they're describing is very, very different and require different solutions. So just to give you an example, the most like what I'll call optimistic model here says there should be about a 3 degrees C increase in temperature globally. So what does that amount to? Well, that amounts to, like, we're going to be experiencing extreme climate events like extreme droughts, extreme hurricanes, and so forth. But the climate will still be kind of recognizable and manageable. Now, if we go up here, some of these models are predicting more than 12 degree C increases. And just to give you an idea, at 9 degrees C, we expect that much of the Middle East will be uninhabitable by people, unfortunately. And that is because if we are seeing that level of increase in temperatures in areas right around the Gulf, we are expecting heat. We're expecting heat events where the temperature will increase to about 140 to 150 degrees Fahrenheit for several days in a row. At that point, people simply cannot physiologically cool themselves down. So they won't be able to go outside. So they'll likely need to migrate, and this will be a massive change. So these are very different worlds that we're talking about here, and they require different solutions. So we need to somehow get these predictions to be more consistent and more accurate. Okay, so how can we do that? So one of the things that we can do is better understand soil carbon dynamics in the future. And that is because soils really contain a lot of carbon, and that can then influence the CO2 in the atmosphere. So here I'm showing you the total amount of carbon in the atmosphere, in the land plants, and in soils. And each of these boxes here is sized according to how much carbon there is. And you can see here the amount of carbon in like dead material in soils is actually twice as much as we see in the atmosphere, and more than twice as much as what we see in plants. Now, the carbon in all these pools, they're connected. So as we have the carbon sitting in soils, we have microbes acting on that carbon to decompose it, and they release CO2 to the atmosphere. All right, now there's so much carbon in the soils that even a proportionally small change in these microbial activities here could add up to a relatively large change in how much CO2 is being released to the atmosphere. So we need to understand these soil carbon dynamics in order to understand atmospheric CO2 in the future. And we know that the Earth system models that we're using are not very good at all at predicting soil carbon dynamics. And I'll show you. Okay, this right here is a study where the co-authors asked, okay, of these Earth system models that we're talking about, how well can they, sort of predict current soil carbon stocks? Even just, that's the easiest thing that we can ask them to do right now for soil carbon is, can they predict current soil carbon stocks? And the answer is no, not very well. Okay, so this right here, this is a map of our empirical measurements of soil carbon. So this is what we can be pretty sure is in the ground. And the warmer colors here have more carbon in them. These right here are 9 different Earth system models, and they're showing how much soil carbon they're predicting right now. So you can compare, like how well do they replicate this actual map? And the answer is no, they don't replicate this actual map very well. First of all, they don't agree with each other very well at all. And second, the just total amounts of carbon are off. And then also where the carbon is in the world is off for a lot of them. And so if you actually look at a like pixel by pixel comparison, the models can only predict about 6% to 39% of the actual variation in soil carbon. All right, so there's a lot of room for improvement here. And that is what we're trying to do in our lab. We're trying to get the information to put in these models to improve the predictions. Okay, so fungi are key to this. That's because they drive many aspects of soil carbon. And I already mentioned their role in decomposition. All right, so we have some fungi that are growing through the soil. They're growing on this dead material from plants and microbes. And as they break it down, they release CO2. Okay. Now, I'm going to talk a lot in this talk about recalcitrant carbon. So recalcitrant carbon is carbon that is generally difficult to break down. All right, so generally this carbon lasts a long time in the soil. It has a very large total effect on how much carbon is stored in the soil. That's why I get so fixated on recalcitrant carbon. Now, fungi, as I mentioned, they can break down this recalcitrant carbon, right? There are fungi that can do that. And so they release CO2 as part of that. Now, there's another thing that fungi do. And as they're growing through the soil and they're taking carbon up, part of that they release as CO2, but then part of that carbon they incorporate into their own biomass, so they grow. Now, some of these fungi, when they're making their own biomass, they're making molecules that are themselves recalcitrant, that are very tough to break down. Okay, so this is a picture of a fungus called Armillaria, and it's been growing through the soil, and it's made these like hyphae structures. And these hyphae structures are very tough. They're basically like woody. And if you were to grab them and pick them up, they would be tough to break. All right, so they're recalcitrant, they're difficult to break down. So when a fungus is making this material and they die, part of these recalcitrant carbon compounds then stay in the soil, all right? And when they're staying in the soil, they're not being released as CO2. Okay, so they're not contributing to potentially soil, to climate change. Now, this may not seem like a very big process, right? We think of microbes as being very small, but it does add up over time. And if you look at organic carbon in the soil, a lot of that organic carbon looks like it originated from microbes. It's the stuff that fungi are putting in their cell walls. So it does add up over time, and it's important. Now we have another fungus that is my favorite fungus, and these are mycorrhizal fungi. So these fungi can form relationships with plants. They're mutualistic. They grow on plant roots. And here you can see a plant root that's being colonized by this fungus. And what they do is they'll grow out into the soil. they'll mine the soil for nutrients like nitrogen and phosphorus, and they'll return a portion of that to their host plant. Right? And in exchange, the host plant provides carbon to them for energy. And through this relationship, they improve plant growth quite a bit, actually, by about 50% when they're growing on plants. Now, the plants are pulling CO2 out of the atmosphere via photosynthesis. And so this is another function that fungi have that affects CO2. But in this case, it's the opposite of decomposition. Okay, so they're drawing CO2, they're causing CO2 to be drawn out of the atmosphere. Okay, so these are three very different functions that fungi can have. And they have very different effects on climate change. So they could either accelerate climate change through this process of decomposition, they can slow it down by improving plant growth, or they could hold that CO2 as storage and offset, slow down future carbon change through this mechanism. Okay, so now the question then becomes is, does it matter which fungi we have in the soils? They're doing these three different processes. Do all the fungi in the soils kind of contribute to all of these processes? In which case we don't care what fungi are in the soils for this reason, it doesn't matter. They're going to be doing the same things. But if different fungal species specialize in these different functions here, then we do end up having to care which fungal species are in the soil. And if we know which fungal species there are, that can help us with our predictions. Right? So we do have a hint that fungal species differ in how much they perform these three different functions. And that is because only a few fungi form mycorrhizal relationships with plants. So already that seems like that's a specialized function. Okay, what about the other two? Are there fungi that specialize in decomposition but don't make recalcitrant carbon, or vice versa? Okay, in that case, we need to consider that as well. Okay, so that is one of the big questions that we are asking in our lab is, do different fungi perform these different functions? Okay, so one of the first things that we wanted to do is just to test whether different morphological groups of fungi might specialize in these different functions that I was talking about. The reason that we started with morphological groups, they're the low-hanging fruit. All right, so we know from natural history, because we can kind of see these fungi, we know that they do kind of differ in their roles in the ecosystem and so forth. So it seemed like it was a good place to start. I think they might be doing different functions in the soil. So here are the morphological groups I'm going to be talking about. First, we have filamentous fungi. These are fungi that form kind of rigid tube-like structures in the soil. These are called hyphae. I'm going to talk about free-living filamentous fungi. These are fungi that don't form relationships with plants. They don't have relationships with other organisms. They're just living by themselves in the soil. And we also have yeast here. Now, yeasts are fungi. When I first started studying fungi, I did not know that because they did not look like fungi. They look more like bacteria, right? So they're unicellular here. They reproduce by budding, but they are fungi. And one of the things about yeast is that they are known to be really stress tolerant. They can live in extreme environments. So they can live in water, they can live in hot springs, they can live on glacial ice, places where other microbes are not able to live. That's where you'll find yeasts. So they're different already. And then we have mycorrhizal fungi, which I already talked about. And here's, I think, also a very beautiful picture of a mycorrhizal fungus. This was like white velvet covered. And these again form relationships with plants. So I might expect them to be acting with carbon differently. So I'm going to talk about a study that I performed with Jay Lennon, where we asked, do these groups differ in their effects on soil C under climate change? Okay, so to address this question, we asked if they had different traits related to these functions. All right, so we needed to find different traits that might be related to these functions that fungi would have that we could look for. Okay, so we found like a whole array of traits to examine Some of them are related to decomposition, some are related to building up recalcitrant carbon in the soil. I'm not going to go through all of them at all. I'm just going to give you a few examples. Okay, so one is a decomposition trait. So some fungi, not all, but some fungi can degrade really tough, really tough carbon, like the carbon that you find, say, in dead wood. Okay, so a lot of the carbon that you see in dead wood, you know how wood is really hard and tends to decompose slowly? That's because it has a lot of lignin in it. So lignin is a very large, complex, randomly constructed compound. And so here is just an example of a lignin molecule that you might find in wood. Or you can see here, it's a mess. All right, so there's lots of stuff going on here, lots of different bonds, a lot of aromatic rings, difficult to break down generally. But some fungi have evolved the ability to construct an enzyme called lignin peroxidase. And these enzymes, they can release them out into the environment and they'll start to degrade lignin. And you can see here, this is a scanning electron micrograph of wood that is being attacked by fungi making this enzyme. All right, so they're breaking down the cell walls in this wood. And this is an advantage for the fungus because once they break down a cell wall, they can grow inside that cell and get all the sugar and nutrients in the cell. But first, they've got to get that lignin. Okay, so lignin is a really important controller of overall decomposition of materials. And we want to know which fungi can do that. Okay, now there's another trait that we can look for in fungi, and that is a trait that allows them to tolerate stress. And there are many, many ways that fungi can tolerate stress. But one of them is to make this carbohydrate. It's called beta-1,3-glucan. And beta-1,3-glucan is a compound that some fungi incorporate into their cell walls. And so this is a cross-section of a fungal cell wall right here. This is the membrane. Right here, this is chitin, which almost all fungi make. And then here are some mannose proteins, which again, most fungi make. But then some fungi that make beta-1,3-glucan, they can add this extra layer of carbon into their cell walls, and it makes the cell walls very tough, right? So it's difficult then for those cell walls to break. And it helps the fungi tolerate freezing, stress, and also tolerate drought, because then the fungi are less likely to lose water. Okay. So this is an important stress tolerance trait. We know which gene encodes for it, so that's very useful. And another thing that I want to point out is that this trait actually has a dual function. So beta-1,3-glucan is very difficult to break down in the soil, right? So we know because when we are trying to extract it in the lab, it's not water soluble, it's not soluble in acid, it's not soluble very well in bases. Likely when these fungi are making this beta-1,3-glucan, and they die, this compound likely remains in the soil, and the carbon in this compound is locked in for some time. All right, so this has a dual trait. Okay, so we wanted to know how are these different traits distributed among fungal species? Do different fungal species have different traits? It's very difficult to measure all these traits all under the same conditions in fungi because fungi are just so different, have such different demands. One of the ways that we decided to approach this question is to look at fungal genomes to see which fungi had the genes that encoded for the traits we're talking about. Now, just the fact that the fungi have the genes doesn't necessarily mean those genes are expressed, but it does mean at least they have the genetic capacity for that trait. Now, at the time we were doing this study, there weren't that many fungal genomes that had been sequenced. There were only about 157 whole genomes that had been sequenced. But luckily, all those genomes are housed in the Department of Energy's Joint Genomics Institute website. And so we could go in and search the genomes of all these fungi and see which fungi had which genes encoding for these traits. And we did find, these morphological groups that we're talking about had very different traits in them. And so here's the examples that I was talking about. For decomposition, it turns out free-living filamentous fungi seem to be much better decomposers of lignin than the others. All right, so just to show you, These are the 3 different groups I was talking about. Here's the gene frequency for lignin peroxidase in their genomes. This is the number of copies of the gene per 10,000 genes. And you can see here that the free-living filamentous fungi generally have many more copies of this gene than the others. And in fact, the yeast basically had no copies of this gene. They simply were not able to degrade lignin. Mycorrhizal fungi also were not great at degrading lignin. So already we know if we see a free-living filamentous fungi, likely it's a better decomposer than the others. Okay, what about stress tolerance, the ability to make beta-1,3-glucan? Well, that also varied a lot between these 3 groups. In this case, yeasts were great. I mean, were much better. They had better capacity for generating beta-1,3-glucan than the others did. All right, so here you can see the yeast had almost 4 copies of this gene in their genome per 10,000 genes, whereas the free-living filamentous fungi and mycorrhizal fungi had much fewer. Okay. So this means that we're basically seeing different lifestyles or suites of traits in these fungi. So on the one hand, you have free-living filamentous fungi that can decompose the recalcitrant material, but they don't seem to be able to tolerate stress very well. Right? And on the other hand, you have yeasts that can tolerate that stress, but simply cannot decompose lignin. All right, so how are these groups potentially responding to climate change? Oh, by the way, mycorrhizal fungi were not good at either. That's too bad. They're my favorite fungi, but they have other skills that I just didn't talk about in this talk. They just happen to not be very good at stress tolerance or decomposition. Okay, so what's going to happen if we have climate change to these different groups? Well, here you have climate, and let's say in some areas, climate change makes the climate more moderate. Well, in that case, we might expect free-living filamentous fungi to perform better because they're relieved from that environmental stress. They will have a stronger ability to decompose recalcitrant carbon, and that may reduce soil carbon storage and release more CO2 to the atmosphere. That could accelerate climate change. On the other hand, if you have an area exposed to more extreme climate, like maybe Southern California, we might expect yeast to respond positively because they have the stress tolerance. They may be producing more recalcitrant carbon because they're making that beta-1,3-glucan, and that could add up to increased soil carbon storage, and that could help reduce maybe climate change in the future. Okay, now these different feedback loops present different scenarios, and we wouldn't have hypothesized them if we hadn't known that these 2 groups had sorted their traits the way they did. Okay, so let's see if this is going to happen. All right, so we tested out this prediction in 2 different ecosystems. One where the climate is expected to become more moderate in response to climate change, and one where it's expected to become more extreme. So the one where we expect the climate to become more moderate is this Alaskan boreal forest here. All right, so just for reference, this is the amount of warming that we've seen already in different parts of the world compared to the late 1800s. You can see here that the boreal forest up here is warming up pretty rapidly. All right, and now before that, it was quite cold. So we expect temperatures to be better for fungi. In Costa Rica, it was already pretty warm. It is warming there too, not as fast, but it is warming. And so we expect that if temperatures increase even more beyond that, some of the fungi may be hitting their environmental limits. Okay, so we went into each of these sites and studied how climate change could affect the fungi. So the first one that I'm going to tell you about is this warming experiment that we did in Alaska. We asked in Alaska, how does climate change alter soil fungi, and what are the consequences for soil carbon? Now, one of the important things about Alaska is that there's a ton of carbon in the soil. It's been adding up there for millennia. And also there are a lot of fungi. Here's pictures of some of the fungi that we see in this site. Okay, so one of the ways that you can simulate global warming is to use greenhouses. Now, I mean, the best way to simulate warming is to actually actively heat the soil in the ecosystem, maybe with heating coils or something like that. But we couldn't do that. We were out in the middle of nowhere in Alaska. We didn't have access to electricity. So we did passive warming with greenhouses. So we, actually my then postdoc, Steve Allison, he constructed these greenhouses here with plastic. And he put them on 5 different plots, and they warmed the soil kind of moderately by about 1 degrees Celsius. Okay. Now, they could potentially interfere with precipitation, as you can imagine. To offset that, what Steve did was he put these gutters on the greenhouses, and then he had tubing that would direct the water that the gutters collected into the plots. And as I'm describing this, I'm feeling really self-conscious. I'm talking to engineers, so we did our best, okay? I know it doesn't look great, but it did work.
Creative. This is good and creative. Yeah, that's exactly what we need.
It worked. It got the job done. So we had 5 warming plots and 5 control plots, and they've been going now for like, I think 13 years. After 8 years of warming, we collected the soils from each of these plots, and we did molecular analyses to identify the fungi in them. And we could use DNA sequencing. Our techniques now are so advanced, we get sequences with no problem. We got about 4,000 fungal sequences per plot. We could identify them to species. We found that they represented about over 1,300 different species. And of those species, about half of them, we could determine which morphological group they belong to. And so we analyzed half of those fungi, and we asked, do these different morphological groups respond differently to climate change? And just to remind you, we expected the filamentous fungi to more positively respond to warming. And that is what we found. All right, so this right here, this is a graph showing how these 3 different morphological groups responded to the warming treatment. Here on the y-axis, this is the warming response of different groups. This is compared to the control treatment. So if you see here, like 30% up here means that that fungal species became 30% more abundant under warming. All right, and here this indicates they became less abundant under warming. Okay, so the free-living filamentous fungi, they increased significantly in response to warming. All right, not a whole lot, just 4%, but they did increase, and this is what we had hypothesized. Now, the yeast, on the other hand, declined, and they declined quite a bit. And this also makes sense considering what we know about yeast and looking at their ability to tolerate stress. They tend to specialize more on tolerating stress, right? And they may have had an advantage back when it was a bit colder, but that advantage might have declined with warming. And so the other fungi might have outcompeted them. And so they were less abundant. Ectomycorrhizal fungi did better under warming. I'm glad about that. And again, they did not have a lot of good stress tolerance. So it makes sense that they would do better under warming. Okay, so now what does this mean then in terms of feedbacks on climate? Well, so this is what we think the Alaska scenario is. The climate's becoming more moderate. The free-living filamentous fungi did respond positively. We expect that they'll have a stronger ability to decompose recalcitrant carbon, and so soil carbon storage might be decreased there. All right, so we asked, does that actually happen? We need to test that. So one of my former PhD students, Adriana Romero Alvarez, she went out and tested that up in Alaska. She made what we call litter bags. These are made from window screen. She collected dead spruce leaves and put them in these litter bags and simply just put them out in these plots and let them sit there for up to 18 months. And then she collected them periodically and measured how much recalcitrant carbon they had in them to see if the recalcitrant carbon turnover changed. Okay. And it did, right? So warming did affect the turnover of recalcitrant carbon in the way that we expected. All right, so I'll explain to you. These are her results. On the x-axis here, we have decomposition time. All right, so she collected the leaves at 4 different times. Now on the y-axis here, we have the ratio in the leaves of recalcitrant carbon to non-recalcitrant carbon. Okay, so that's how much is remaining. So first I'm going to explain the pattern that we found in the control plots. Okay, so let's look at the control plots. As decomposition progressed, the amount of recalcitrant carbon compared to non-recalcitrant carbon increased. over time. Okay, that's in the control. Now that's very, very common. And what's happening there is that the microbes are degrading the non-recalcitrant carbon preferentially. Okay, so that means that the recalcitrant carbon makes up more of that leaf litter, all right, at that point. All right, so this is very common. Now, the warming scenario had a different response. It's more unusual. So here, basically, this ratio did not change over time significantly. And so what's happening here is that the fungi appear to be attacking the recalcitrant carbon as much as they're attacking the non-recalcitrant carbon, because they've shifted in their preferences from avoiding the recalcitrant carbon to breaking it down. All right, so it looks then like this recalcitrant carbon is going to be turning over more quickly under the warming conditions. And again, that's consistent with our hypothesis and how we see the fungal community changing. Okay, so that's the story for Alaska, where climate should become more moderate. Now, what about the Costa Rican tropics? Okay, so the Costa Rican tropics that we study are seasonal dry forests. So these are forests that do have this dry season where it gets quite arid. And if you go there in the dry season, it looks a lot like Southern California in the dry season. Right? A lot of the trees have lost their leaves, the grasses are dry, and so forth. Okay. Now, if you go up the mountain from these seasonally dry forests, you start entering cloud forests. And in cloud forests, it's a different setting. So in cloud forests, we have clouds that form during part of that dry season, and they're a source of water. So in cloud forests, the drought is basically buffered. Now, it appears like this is a better, these are better set of conditions for fungi. If we look to see, compare these two sites, the cloud forest and the seasonal dry forest, the cloud forest has higher fungal biomass and more fungal activity. All right, that makes sense. Fungi need water, just like most organisms. Okay, so the problem is that climate change is actually lifting the clouds off these mountains. All right, as the temperature of the atmosphere warms, the clouds are forming, but they're forming higher and higher up, so they're no longer touching the mountaintops. So now these cloud forests are starting to look more like seasonally dry forests. Okay, now what's going to happen with the fungi there? Well, my former PhD student, Caitlin Luby, asked this question, and she studied it in Monteverde cloud forest in Costa Rica. Now here, we actually weren't even able to construct greenhouses. These forests are a lot more fragile, and it's very difficult to get permission to make an experiment like that. Instead, Caitlin took advantage of a climate gradient by going down the mountain, right? So here's the deal. So here's Monteverde. Monteverde is at the top of the mountain here at about 850 meters elevation. As you go down the mountain, that's when it starts getting warmer and drier, and then you get into these seasonally dry forests. So what you can do is if you stand on top of the mountain here and take a hike down, you're essentially going forward in time in terms of climate change. You're experiencing a future climate that the cloud forest will experience. So what Caitlin did was she took the fungi with her. She got the fungi up at the top of Monteverde cloud forest, and she brought them down this gradient to expose them to these warmer and drier conditions. Right. And so she set up a set of sites that varied in elevation, and they did get warmer and drier as you go downhill. So now she needed to transfer the fungi from that top site down to the drier sites. Now, I'll just tell you, it's difficult to manipulate fungi outdoors. They are small and they get away from you. So what we did was we made what we call fungal cages. So fungal cages are like those litter bags that I showed you before, except they're made of a nylon membrane with very, very small pores. The pores are just 0.42 micrometers diameter. So this means those pores are too small for fungi to grow through. So if you put a fungus in this cage here, they're trapped in there. Now, the nice thing is the pores are big enough for water and nutrients to pass through. So the fungi are still experiencing the environment around them. They're just trapped in these bags. So Caitlin then, she collected soils from the Monteverde cloud forest, the top site. She put the soils in these bags and then transferred them to different sites downhill. And here's one of the sites. Okay, and here's the experimental design she used. She picked 4 sites downhill from the cloud forest. These sites vary in soil moisture. And when we looked at the data later, it seemed like soil moisture was the main driver of changes. So I'm going to focus on that. So here, compared to this top site in the cloud forest, these sites either have a decline of 5% moisture down to about 20 or 21%. Okay. So she took bags from this top site. She put them in these different 4 sites. And then she also took bags at the top site and just kept them in the top site as a reference. Okay. So she waited about a year after she transplanted the fungi, and she collected the bags again, and she saw, okay, which fungi became more abundant, which became less abundant after the transfer. Okay, and we found that the fungal taxa in those cages really differed a lot in their drought tolerance. So I'm going to show you a couple of examples. So here's a Laetoporus species. Here's a picture of it. It was relatively abundant in her cages, and she found that it seemed pretty drought sensitive. All right, so here is the degree of soil drying that the bags experienced compared to the reference site at the top of the cloud forest. And this right here is the relative abundance of the species in terms of number of reads in the community, number of DNA sequences. So right here, this is kind of the cloud forest right here. This species is doing relatively well in the cloud forest. And as soon as you take it out of the cloud forest, it just drops right out. It cannot tolerate that drought. So we call it drought sensitive. Okay, in contrast, here's another species. This is a Cryptococcus species. It's a yeast, right? It's really, really abundant in these bags. And it actually had the opposite response as Laetoporus did. It actually increased in response to drought. And so you can see here, it's pretty linear too, right? So we call this a drought tolerant species. All right, so we could go through for each of the species that Caitlin found in her bag. We could quantify how drought tolerant versus how drought sensitive they were, right? And basically we did stats for each species. We calculated what's called a correlation coefficient. Basically, the higher this number is, the higher the R value, the more this abundance went up with drought. Okay, and if there's a negative number, that means that the abundance of the fungus declined. It was sensitive. All right, so we calculated these numbers for every single species she found. And there we go, that's the index. And she actually altogether found 309 different genera of fungi that she was able to calculate the drought sensitivity of. All right, and here's what she found. These fungi really did differ in their drought sensitivity based on their morphology. Okay, so here's the yeast in the bags, and here are the free-living filamentous fungi. And here is their drought response in the Costa Rican forest. So these numbers here, the higher up you go, the more drought tolerant the groups are. And you can see here the yeast actually increased significantly in response to drought, right? And in comparison, free-living filamentous fungi didn't overall have a big change in response to drought. Okay, so overall, we expect that if we have drought in the cloud forest, that will select for yeast. Okay, so what does that mean? Well, are the yeast then, do we expect them not to be such good decomposers so that we will have less turnover of carbon in these sites in the future? All right, well, we could figure out if these yeast were likely to be good or bad decomposers and so forth by going back to the whole genomes of the fungi that we found in the cloud forest. Again, we have a whole genome sequence. We can look and see what kind of genes they have. And we were lucky. Caitlin was able to find whole genomes for 37 of the genera that she found in the tropical forest. So we could ask for each of those genera. We know what the drought sensitivity of that genus is. what's its ability to break down recalcitrant carbon? Right, we actually found that there was a relationship between these two traits in the fungi we found in Costa Rica. So right here, this is drought response in the tropical forest, what I've been talking about. Again, this means if you've got positive numbers here, this means that the genus was tolerant of drought. Negative numbers means that it declined under drought. Each of these symbols here is one genus from the fungal cages. And then this right here value is the number of lignoperoxidase genes in the whole genome for that genus. And you can see here that there's a negative relationship. So these fungi that are particularly drought tolerant over here, they have very few copies of lignoperoxidase. They're just not good decomposers. Okay, on the other hand, the fungi that did have a lot of copies of lignoperoxidase that could decompose that wood tended to decline under drought. Okay, they just didn't do very well. Okay, so overall then we expect then to have a feedback in the tropics that looks very different from what we found in Alaska. In the tropics, we expect more extreme climate. We know that selects for stress tolerators, and we might expect those stress tolerators to not be very good at decomposing carbon. So we might expect there to be more recalcitrant carbon in the soil over time. Now, just to remind you, that contrasts with our Alaskan responses, where we expect a more moderate climate should increase decomposers in the ecosystems and might reduce recalcitrant carbon in the soil. So two very different feedbacks happening in these different regions. Okay, so this is all something that we can then incorporate into these Earth system models. All right, so how do we do that? Well, current Earth system models don't even simulate microbes, because they don't even have microbes in them at all. Instead, we're turning to next generation models that are built from the ground up to include microbes. These next generation models can actually include traits, the traits that we've been talking about in their simulations. All right, so they can generate a simulated community of fungi where they have these different trait relationships, where you have fungi that are good at warming, that can do well under warming, tend to have a better capacity for decomposition and so forth. We can initialize a simulated community with that suite of traits, then expose them to climate change, see how that community sorts out and what sort of functions they have. We're early on in this endeavor, but so far it looks like these models are doing a better job at predicting soil carbon cycling than the sort of classic models. And so we're very optimistic that this is going to be improving our predictions of climate change in the future. So overall, what we found is by looking at the traits of these fungi and seeing how they sort out between groups, we can help improve predictions of climate change. And then now we're able to put these in these next generation models. And we hope to, over time, really start to get models that agree with one another and are more accurate in predicting climate. We're excited about these prospects. And so just to thank everybody, thank all of my collaborators and funders. Thank you for listening. I really appreciate it. And also thank the fungi for being great to work with. They're always great to work with. All right, thank you. And I'm done now. And if we have time for questions, I'm ready for questions.
Thank you. I don't know how to clap, but this is the place to clap.
Thank you.
Oh, excellent presentation. Yeah, no, very, very, very exciting work. I think some students have questions.
Yeah, and I have to apologize. I wasn't able to follow the chat.
No, that's okay. We just
That's okay. We can read it out for you.
Okay, all right.
Yeah, go ahead, Ren, if you want to ask.
Yeah, thanks, Professor, for your presentation.
Thank you.
Yeah, I'm wondering that because in the soil environment, there might be symbiotic interaction between fungi and bacteria. So that change of the fungi in this changing environment is directly from the environmental factor, or that might be from the bacterial change?
Yeah. Oh, that's such a good question. So I completely omitted the bacteria in this talk. So the bacteria are also responding And they were part of those cages that we talked about. They're in the pots in Alaska. They certainly are changing in their community composition, and they have a whole different suite of traits, and they may have different trade-offs. So for the bacteria, for instance, we haven't seen as strong a trade-off between stress tolerance and decomposition. So they may be able to buffer that change a bit. Now, you're asking about interactions, and there are a lot of interactions between fungi and bacteria, and they can be mutualistic. So there are these bacteria that we call helper bacteria because they tend to grow right alongside the fungi. They're often attached, actually, to their extremities, and they will provide like nutrients and other things to the fungi, and then the fungi also exude sugars to feed them. And so that's just one example. There are many others. They likely compete too. And so we haven't included that in these studies. And I think that's something that we really are working on now. We're creating artificial communities where we manipulate both the fungi and the bacteria, and we're putting them out in these environments to see how the interactions can affect them. Thank you.
Okay, thank you.
Any other question from students? I have questions, but I rather have you guys ask questions.
Students, yeah.
Yeah, the nice thing is that like you don't really know very much about fungi, so you can ask any question. It's totally valid. As long as you're willing to have me say that I don't know.
This is a science project question. I went to Home Depot 2 weeks ago and I saw this growing mix. They say that, I took a picture. It says sunshine growing mix with mycorrhizae.
Oh yeah.
That means they are claiming that they have all the fungi inside that mix. So if I buy that compost and put in the, let's say, stormwater treatment system, does it mean that any plants over there will somehow automatically find ability to work with this fungi, or they will just not?
Yeah, I can actually give you a solid answer on that because we've done a study on it. So we have looked at we're not doing bioremediation in our ecosystems, but we are trying to restore them here. So we have ecosystems where we're trying to help the native plants grow. And so for instance, we asked, like, if you get a commercial source like that with the mycorrhizae fungi and you add it to our ecosystems, it hurts them. So the plants do worse. And that we think that's because there are mycorrhizal fungi in there, that's for sure. But mycorrhizal fungi differ too. They're on a spectrum. And some of them are actually not that helpful for the plants. They just grow on them and take their sugars and don't do anything. And those are the ones that are in the commercial mixes because they're the easiest to grow. On the other hand, if you take soil from an ecosystem that is in good shape and seems like the plants are doing well, and you put that in an ecosystem, then that helps the plants grow a lot better because those mycorrhizal fungi are native and they're actually adapted to those plants and can deal with the environment. So I generally say, don't bother with the commercial inoculant, like
just
snake
get
oil.
healthy soil and mix that as inoculant.
Just it doesn't take very much, like a handful or so. Just mix it in. It's free.
Oh, that's interesting. So that means, that's another question I was thinking is, you said that fungi have really good ability to take nutrients for plants. So if the soil has already a lot of nutrients, which is the case in many stormwater treatment systems because they capture nutrients, do you think that this abundance of fungi or mycorrhizal fungi will decrease because of
They do? Yeah, and I can answer that too because we tested it. If you add a lot of nitrogen or phosphorus, both of those, then that will knock the mycorrhizal fungi out. And the idea there is that the plants, they only have so much carbon that they're dealing with. And if they have plenty of nitrogen and phosphorus, like why invest that carbon in the fungi? They invest it instead in fruit or leaves or something like that. So yeah, so if you're trying to use the mycorrhizal fungi for remediation, but there's a lot of nutrients in there, like you have to find a fungal species that is good at that remediation, but can still be maintained under the high conditions. It's stressful. It's actually stressful for the fungi.
Yeah, I was going to talk about, so we emphasize so much on the carbon cycling, but then there's this whole role of of nitrogen with respect to growth and, selection of fungi, but also the nitrous oxide and nitrogen gases having an impact on climate change. So it just gets really complicated.
Fungi make nitrous oxides too.
Yes, they do.
And so I just talked about, you're right, I just talked about the carbon part of it. There's like whole other cycles that they're involved in. That's for sure.
Yeah, and they feed off of each other. So it's important for us to understand with respect to ecosystem services, remediation, but also agriculture.
Exactly.
So.
Yeah, even like heavy metals. Some mycorrhizal fungi will actively absorb. They'll actively forage for and absorb heavy metals, and others will die if they're exposed to them. So yeah, it's pretty cool.
Yeah. So I wanted to convey my colleagues Jenny J's message that she lost connection, but wanted to say that your seminar was very, very exciting and very clear for this audience. I know it takes a lot of work, of course, to do such solid science, but also to be able to explain it to a whole diverse audience. And we really appreciate it. And I hope the students learn not just the content, but also the communication skills.
Thank you. And I see a young scientist is online here. So I hope that she...
We were talking about like policy work, and I was wondering if you would elaborate on that a little bit.
Yeah, so I've gotten really involved in local actions here, and I'm actually on my city's Green Ribbon Environmental Committee. So we advise our city council on policies and things. And so I was telling Sanjay and Shaylee earlier that a big part of Irvine used to be part of this Air Force base. And when they were maintaining the planes, they'd haul the planes out onto the bare ground and then just douse them in solvents. And then those solvents went into the ground and they are still there. And so a big part of Irvine is like on this, it's called a plume, the El Toro plume. And it's, first of all, it's a social justice issue because they're not able to use their groundwater. And second, it ends up being a race issue because those plots were sold preferentially to people of Asian American backgrounds who had just immigrated and didn't quite understand what had happened there before. And so we're really trying to work hard to deal with this plume issue. And I am interested in how fungi could be relevant, but I don't have the expertise in it. And so I'm really glad to be able to connect with this group here. I think that there's a lot of opportunities, and I imagine that what's happened in Irvine is not rare, and there's these issues in many places. And so I can see how it's so important.
Yeah, so El Toro plume is primarily chlorinated solvents. And then that's the DoD project I was talking about. My student Ivy Kwok is here. Rain, who asked the question about bacteria, he's a current postdoc working on, again, all the omics and trying to make the GMO that can, probably degrade a whole bunch of contaminants all at once.
Yeah.
So real exciting part of research. Others are here too. Pia's here. I think Alex dropped out. But yeah, my group is a fan of your work, and they've been reading a lot of your papers, trying to catch up on fungal biology.
Well, that's great. Well, once we start getting back into the real world, they're all welcome down here. They can come down and talk with us or give presentations. Or we're very happy to have that connection.
Yeah.
Thank you. This is great. I'm glad that you accepted, because for a long time I've been thinking to have you here.
Yeah.
So this is
great. Well, yeah, I'm glad you reached out. That was a lot of fun. It's good.
Yeah, I'm going to stop recording.